I'm very proud to introduce Dr. Stephen Cowen from the Department of Psychology, and he is going to tell us about the science of hallucinations, how our brains create reality, and how some drugs affect our perception of reality. So a big round of applause for Dr. Stephen Cowen.
Thank you. Thank you very much. It's always a good sign when you're going to give a talk and you spill half a glass of water over your front shirt. So with that, I'll begin actually giving a little introduction about who I am. As Shepard mentioned, I'm a scientist over in the psychology department. I work near the hospital, and I study brains. I study how our brains make decisions, how Parkinson's disease affects the brain, and how we might develop tools to help solve some of the symptoms, or at least resolve some of the symptoms of Parkinson's disease. Those are some of the things I work with. But before going further, I want to give a little background as to how I got here. I think usually sometimes people are curious as to how a scientist becomes a scientist. At least I like to tell this to some of my students to say you don't have to go the traditional route. So my degree is actually in business. I had a business degree from the University of Wisconsin-Madison, studied there for a while, and then went from there directly into the Peace Corps, into a small island in Honduras, and worked there installing solar panels and teaching communities how to manage micro businesses. But that time away kind of gave me pause to think about what I wanted to really do with my life and reflect on how much I just always have loved science. Science has always been this kind of grounding force from back as far as I can remember. And that time away kind of pushed me back there. And I knew I wanted to study the brain, I wanted to study the mind, but I didn't know how. So I took a number of courses in philosophy and neuroscience, which was the stellar course, the one that turned me on, and psychology and other courses too. And then I eventually found my way back to Arizona, where I went to grad school. I then moved to San Diego after getting my PhD and ran a laboratory there for a little over 4 years and learned how to surf, which was great. And then, luckily, found my way back here. And now have a great lab and some great students who are actually out here in places. So, hi, students. And out there in the audience. And some great colleagues, too, who actually made it this way. So, and you'll actually maybe hear a little bit about them in the talk. All right. So going back to what in my lab studies, one of the things we work with or study is decision making, and we're all making decisions. Even these guys, these javelinas that are tipping over our garbage all the time, are making deep, meaningful decisions. They're trying to evaluate the costs and benefits of tipping over that green garbage pail right before I have to get to the airport at 5 in the morning. Or just going and munching on a cactus, and usually they pick the garbage pail. So I'm very interested in how that cost-benefit decision-making process goes on in the brain. And we think a lot of that is happening here in the frontal cortex, this frontal part of the brain. And that's convenient because we believe the origins, at least of many of the components, at least of hallucinations that we have, whether drug-induced or not, occur because of dysregulation, dysfunction of this frontal part of our brains. And that'll come up a bit in the talk as well. Yes, psychedelic cats come from the frontal cortex of the brain. All right. So going back to Parkinson's disease, and another reason what kind of connects me a little bit with hallucinogens and hallucinations and how the brain produces these things, is in the process of working with my collaborator, Dr. Torsten Falk, over in the neurology department. He and his colleagues came across a novel application of an age-old anesthetic called ketamine, which you probably have heard of before, that can actually also induce hallucinations, but at high doses puts you out so that you can get an operation. It's still used today in certain special circumstances, but one potential benefit that Dr. Falk and others are working on is that this drug may help limit or reduce some of the negative symptoms of Parkinson's disease. At least when you take Parkinsonian medication over and over, you develop these tremors. This might help reduce some of those tremors. And some recent research suggests that ketamine itself might actually also help with things like severe depression. So that's another reason that connects me, I guess, to a little bit with hallucinations. Not because I'm an active participant in this research or user of hallucinogens, but I do study the chemicals and the brain correlates of these chemicals in my lab. All right, but before we begin, let's begin with the brain and maybe give a little review of how brains work, just to get you primed up so that when I talk about receptors and neurotransmitters and action potentials, that we're ready for it. We're ready for it. All right, so brains are beautiful things. They're just absolutely gorgeous. We have about 100 billion firing neurons in our brains, each one of us. And those billions of neurons have tens of thousands of synapses connecting them. So it's this incredibly complicated system, beautiful system in our brains. And these synapses, these connect neurons. So synapses are those little junctions that connect one neuron to another neuron, and they release chemicals when they're trying to communicate called neurotransmitters. And those chemicals are released from things called vesicles, which are essentially fatty bubbles. So a lot of the brain can be reduced to fatty bubbles, really. It comes down to fatty bubbles. And dirty stuff in fatty bubbles. So one of the fatty bubbles are these what we call vesicles that contain the neurotransmitters, but our cells themselves are essentially fatty bubbles that are filled with proteins that receive information, that can detect chemicals, or that can release chemicals like this. But they're not just fatty bubbles filled with chemicals. They're also very sensitive to electricity. And so our brains are what we call bioelectric. They're activated by electric currents, and they're also activated by chemicals that transfer between cells. The electrical part is very interesting to me because that's the majority of the focus in my laboratory, is trying to understand how electrical signals transferred from different neurons to different neurons might encode information about where an animal is, what he's planning to do in the future, what he's remembering from his past. So we're trying to decode essentially the electrical pulses from neurons in the brain and help us understand how we remember and decide. All right, so going down, though, to the drugs, because ultimately we're going to talk about chemically induced hallucinations. And so drugs work in the brain by essentially binding with what we call receptors on neurons. So a receptor is just essentially like a game of Tetris. So you might have a neuromodulator, a neurochemical like dopamine or serotonin or ketamine or some other drug. And what it does is it finds a way to nestle into a receptor on a neuron. And when it nestles in, it causes that receptor to change shape, do some sort of change, configuration change, that can trigger, for example, a cascade of chemical reactions in the neuron, or open a channel to let ions into the neuron, which might cause electricity to happen. Okay, or we'll talk about that more in a bit. And so these receptors and chemicals work together to essentially alter neuronal activity. Yeah, think of it a big game of 3D, really complicated Tetris. That's what most of it, and fatty bubbles. If you remember something from this talk, fatty bubbles and 3D complicated protein Tetris. All right, so I'm talking right now about individual neurons and chemicals, but our brain is composed of 100 billion of these neurons working together or not, depending on your state of mind, to do things. And so what happens when neurons start to work together? Well, as we know, these neurons are chemical and electrical, and so because they're electrical, lucky for me and my research and my students, we can measure these electrical pulses with small antennas or electrodes that we can either put on the surface of the brain or deep into the brain. And what you can measure are brain waves, which is essentially just the organized, orchestrated activities of many neurons at once that can then be picked up, just like you'd pick up a radio wave, for those who still use radios. All right. So there are different flavors of these waves, and we give them different fancy Greek names just to make us feel intelligent. So they go from like delta, theta, alpha, beta, gamma in order of speed or order of increasing frequency, where usually, this is a way oversimplification, but often the slower waves are associated with things like sleep or kind of quiet wakefulness. The faster ones are often organized or associated with more fast processing, waking, and attention and behavior. Everything changes, though. There's far more complication to these different brain waves than would be indicated by this. And we'll get to that in a bit, too. But in general, when neurons work together, when they're sending signals to other groups of neurons, and they're doing it in a coordinated way, they produce these measurable brain waves that we can measure with electrodes in the brain or on the surface of the brain. All right. Now that's about it for the basic neuroscience primer. Now back to hallucinations and how the brain may or may not produce hallucinations. And so I can pretty much well, let's just start with some definitions. What is a hallucination? At least according to this definition, first thing I found on the internet. All right, an experience involving the apparent perception of something that is not present. Okay, well, what does that mean? Let's resort to an internet meme to resolve this. I don't know if you know David. I saw this video. I'm not going to play the video, but David had received or had some teeth removed and went under a general anesthesia and came out a little loopy. And so while he was a bit loopy in the back of the car, he said, "Oh, I've got 2 fingers. I've got 4 fingers." And he was a little crazy, but he asked, "Is this real life?" And it's a deep question. I mean, that's a very deep question. Now, but this video was watched 132 million times. And one. Oh, yes, excellent. Okay, great. 132 million and 1 times. And so, yes, David does ask, is this real life? Well, the answer is no, absolutely not. You're not observing me as I really am. I mean, think of it. A hydrogen atom, these atoms, any proton, is 99.9999999% emptiness, empty. And so we don't perceive reality as it is. So no, this is not real life. We're all constantly hallucinating. We're living in a reality constructed by our brains constantly. And we can even think about this when you think about how even our eyes work. In the middle of your eyeball, in your visual field, even as you're looking at me right now, there's a black hole. A black hole you don't even perceive. There's essentially this hole in your eyeball where all the blood vessels and essentially axons from neurons have to go through in order to get to the back of your head. Well, you don't perceive it, but it's there. And you can do some clever little things that can show you that you have a blind spot, but we don't perceive this blind spot because our brain is actively reconstructing reality. We're creating this hallucination of something that's not there all the time. And there is no white triangle, that too. So we're actively creating the world all the time. And we do this all the time when we sleep as well. Of course, we all have weird dreams. I have very weird dreams, very, very weird dreams. So yes, that's not one of them. All right. But yes, we're always creating very strange realities in our heads. So hallucinations are not anything new and nothing that has to be drug-induced. But of course, we're here for the drug-induced fun part because that's one we hear about and made famous by people like Timothy Leary and all those. And so why not talk about the chemical kind? And because looking at the chemical kind might tell us a little bit about the non-chemical kind as well, and vice versa, by understanding how we might hallucinate during dreaming or during just normal waking through visual illusions. We might learn a little bit more about how our brains construct the world, which is pretty cool. All right, so the classic hallucinogen, the most potent neurochemical or drug that can affect neurons on the planet. So the typical one that people associate with that, the most powerful one is LSD. This is the LSD molecule spinning around in empty space right there. Very, very, very powerful. It was discovered essentially by Albert Hofmann in 1943. Actually, he discovered it a little bit earlier, but he didn't know what it was until he accidentally got it on his fingers and then was taking a bike ride home and started tripping out and flipping out. He worked for a drug company, Sandoz, one of the largest drug companies on the planet at the time. It's still, I think, it's a very large drug company. And he got home and just started really wigging out. I think he thought the person who was cleaning his house had horns, and he had all these strange feelings. And what was kind of fun is he went back to the lab and tried to desperately convince the executives there at Sandoz, one of the largest pharmaceutical companies on the planet, that he found something amazing, something that had these incredibly intense psychological effects. And they said, yeah, whatever, Albert, go home. And he convinced them to try it. And so they did. And so some of the very first users of LSD were high-powered executives at the world's largest pharmaceutical company. And they definitely believed Albert after that experience. All right, here's a little quote by Albert, since he's the one who discovered it. Why not? Little by little, I could begin to enjoy the unprecedented colors and plays of shapes that persisted behind my closed eyes. Kaleidoscopic, fantastic images surged in on me. alternating, variegated, opening and then closing themselves in circles and spirals, exploding in colored fountains, rearranging and hybridizing themselves in constant flux. So yes, he had a very, very vivid experience. And people are trying to be a little more quantitative about the experience of drugs like LSD. And so here's a nice little chart that kind of describes the constellation of feelings engendered by a drug like LSD. Very high on this chart over here are these visual alterations, as you might guess from that description before there. Audiovisual synesthesia, where you combine sound and vision together to hear sounds and things like that. And vivid imagery. A little bit of disembodiment, where you feel a little out of place. So this is actually psilocybin, which is very much related to LSD. But LSD and psilocybin aren't the only hallucinogens people take. There's also other ones, like the one I was talking about earlier, ketamine. It's interesting to compare the effects and the experiences of these two very different chemicals on how we perceive the world. And so ketamine was actually developed in the 50s as a horse tranquilizer and became very popular in certain crowds for other reasons. And it also causes different hallucinogenic effects, but it activates very, very different neural systems and very different receptor systems. Here's one person who said, when I opened my eyes, I could see little animals forming on the pattern of my curtains, which are cow print, okay? Maybe not good taste in curtains, but interesting. All of a sudden, one of these animals frowned an evil frown at me and bore fangs. So not a good ketamine experience, apparently. But that's one of the personal experience of ketamine. And here's that plot again, but you can see there's a little bit different constellation, where there's much more disembodiment in the ketamine experience and a little more impaired cognition and control, but anxiety is way low. So ketamine was actually very reduced anxiety, or had very low anxiety scores compared to LSD. Seeing them by side. Well, I mean, LSD had a relatively low as well, but you can see there's more of a bias towards this disembodiment and impaired cognition. And so we have two different hallucinogens activating and causing some somewhat different perceptual experiences, but they also have some very similar perceptual experiences, in that you do still get these visual alternations. You definitely get vivid imagery in both of these. But what's very interesting is that it's hard to imagine two drugs that are more dissimilar in their structure and in the receptor systems in the brain that they activate than ketamine and LSD or psilocybin. Very different structurally, and they do very different things. So LSD and psilocybin, they activate essentially receptors within the serotonergic system. That's their primary mode of activation. And so they turn on these receptors. We'll talk about this in a second. Whereas ketamine acts principally by blocking a channel that lets ions into neurons, and sodium and calcium ions into neurons. And ketamine can act very, very quickly to cause a rush of ions into the cell, or essentially prevent or block channels that allow rushes of ions into the cells. Whereas these LSD or psilocybin act to essentially alter very indirectly in a more of a chemical cascade the activities of these other types of receptors in the brain that are very sensitive to this. And we'll talk about this in a second. So very, very different drugs acting at very, very different receptor systems, also both producing hallucinations. And so what I want to try to bring out in the talk is how they're similar, though, in many ways, and how they're different on the neural level, in the brain level. All right. So it's often easy to stop a talk to say, okay, hallucinations are essentially produced in the brain by alterations in the serotonin system, or changes in the glutamate system, or increase in activation of a particular region of the brain, and end it there. But those explanations aren't very satisfying. It's like saying, here, I'm going to explain to you how cars work. They work on gas, and then stop there. I mean, it's true. I mean, gas is an important part of how most cars work. Maybe not this car, but how most cars work. But it doesn't really give you a satisfying explanation of the mechanism involved in this case, hallucinations. You need to dig deeper to understand the entire system. If I told you that cars work by exploding gas, you still would probably have a hard time building your own car. But if I understood the workings of a piston and things like that, then I might be able to build it. And so trying to dig deeper into how hallucinogens work at more of a higher level, at a circuit level, All right. So let's get there, start getting there. So with the hallucinogens that are that are activating the serotonergic system, the serotonin neurochemicals, neurotransmitters in the brain, they seem to act primarily at a very, very specific type of receptor in the brain. A receptor, again, are these little, essentially, pieces or proteins on neurons that are waiting for their ideal matched pair. And so serotonin activates these receptors, and LSD and psilocybin also activates these receptors directly. Ketamine only very, very vaguely or very weakly activates those receptors. And so serotonin acts on a particular flavor of these receptors. LSD activates a very specific subtype of the receptors, and this is where the brain gets really complicated. So you might say your brain is sensitive to serotonin. Well, you have at least 15 separate serotonin types of receptors that live in different parts of the brain, on different types of neurons. It gets extremely complicated in there. But there's a specific type called the 5-HT2A receptor that LSD and psilocybin really like to bind to. And what happens is that if you block that receptor, you find a chemical that blocks it, and you take a hit of LSD, you don't get the hallucinations. So there are ways to block that experience by specifically targeting that receptor. Also, antipsychotic drugs may actually work on those receptors by blocking those receptors. And the intensity, the self-reported intensity of a hallucination correlates with the degree to which drugs chemically bind with those receptors as well. So we really think that the serotonergic system and these 5-HT2A receptors are very important for at least the disembodied feelings you might feel, or the hallucinations you might feel when you take LSD. And the serotonin system, just to give you a little overview, is a neuromodulator system in the brain. And by that I mean it's serotonin is a chemical, a neurotransmitter, a neuromodulator that's produced in a small group of neurons deep, deep in the brain. But these neurons send their axons all over the brain. They send it to the thalamus, this kind of relay center in the brain. They send it to the basal ganglia, an area involved in reward and motivation and addiction. They send it throughout the cortex. And so this chemical, serotonin, in a normal brain is going everywhere. And these receptors are also almost everywhere. But they're highly concentrated in the frontal cortex, and they're highly concentrated in an area back here called the posterior parietal cortex. All right. So we're getting a little closer to mechanism. We're getting a little bit closer in that we're identifying a little bit with the regions that are activated, and we're identifying a specific receptor that might be important for the hallucinogenic experience. All right, but going a little bit further, how might the networks, the organized activities of these billions of neurons in your brain contribute to hallucinations? One idea that I like, and I would say is relatively popular in the field idea, is this. Well, I'll call it the isolationist hypothesis. It's this idea that what hallucinogens in general do is they isolate different brain networks inside you. So, for example, you might have a brain network involved in a certain type of memory. You might have another brain network involved in high-level decision-making, another brain network involved in visual perception. When they're disconnected, they're left to their own devices. And when you're left to your own devices, you kind of make stuff up. And so this is one of the general ideas as how hallucinogens may work. You're finding ways, or these chemicals are finding ways to functionally disconnect different brain regions. And you could do this in two general ways. This is some of the ideas that we'll talk about. One is you turn off these central relay centers in the brain. Areas that we'll talk about called the thalamus, or there's another region called the default mode network. That these are areas that interconnect different regions of the brain. If you target and turn those off, then the individual regions that might be involved in decision-making, planning, working memory, and things like that, again, they're left to their own devices, so they can, again, make stuff up. The other way is to just increase the amount of self-talk essentially within those regions, encourage the neurons within regions to not communicate with that guy down in California, but to just communicate with other people here at Magpies, and so to talk more locally. So don't necessarily disconnect, but cause the local people to chatter more with each other. That's another way that hallucinogens may work. All right. Start with the thalamus. The thalamus is this central part of the brain that we often, and we oversimplify it, but we often call it the relay center of the brain. It's an area that kind of routes information, sensory information, motor information, information about planning, memories, and things like that, from one region of the brain to another region of the brain. And what's interesting is that both psilocybin, related to LSD, and ketamine, despite being very chemically different from each other and activating different receptor systems, both dramatically reduce the activity in the thalamus. As a result, that's one way that communication between distant brain regions might be shut down. And so this is what we call an MRI signal right there, and the blue just means the regions decrease. And this is the thalamus right in the middle of the brain there, and the blue just indicates that particular region's decreased. And that's in a human. Now, the work in the rats, at least all that I could find, was done. Sorry, the work on ketamine was all that I could find was done in rats. And here you have a control prior to ketamine, and then here's after ketamine. This is the thalamus in the rat. This is the thalamus in a human. And you can see that in the control, which is probably just saline solution, there's a lot of activity in the thalamus. Afterwards, ketamine pretty much shuts it all down. And so the general metabolic activity in the thalamus goes down with both of these very, very, very different drugs. Now go back to the decider, the frontal cortex, that area that we think and that I study in the context of decision making and planning. This area as well seems to be very important in deciding and planning and predicting the future. And the old story, which has been changing in the last couple years, but the old story was that in general, hallucinogens, regardless of what type, increase activity in the frontal cortex by activating those serotonergic 5-HT2A receptors. And so the frontal cortex starts to talk to itself more. And because it's talking to itself and not listening to anybody else, it starts to make stuff up. It's like a political cabinet, you know? They start listening to their own words, and they say, hey, I must be right. Everybody likes what I'm saying, and so I'm going to keep making stuff up, and we'll see what happens. And so it's kind of like that general idea is going on. You disconnect yourself from the rest of the world, and you just start chattering to yourself. The news story is that it's more complicated. Yes, there might be a lot of self-chatter going on, but this general idea that overall frontal activity increases in general, and that's the common operation of all hallucinogens, is not quite true, because when people actually do more detailed studies of particular regions within the frontal cortex, or even look at the different effects of ketamine or things like psilocybin, they find that there are incredibly different patterns of activity within the frontal cortex itself. So that, yes, all of those very different personal subjective experiences you might have on one drug versus another may be explained perhaps by the specific pattern of neurons firing or turning off within the frontal cortex. So, for example, in one case, you'll see in general that this area right in the middle, called the ventral medial prefrontal cortex. Actually, it's a little bit deep. It's right kind of a little bit above your eyeballs. Very important in emotional processing and some kind of emotion-laden decision-making and cost-benefit decision-making. That area actually can get a little bit more activated when you might be taking, in this case, LSD or psilocybin, in, I guess, that particular example. But other areas like the cingulate cortex, it's a little bit further up in kind of the frontal medial cortex. That area is a connector region, and that area of the frontal cortex actually shuts down quite a bit. And so there's much more complication in how things interact within the frontal cortex when it comes to drugs like ketamine or psilocybin. The other thing that's really interesting is that, in this case, psilocybin, when you're taking psilocybin, it doesn't just cause regions to shut off or to disconnect from each other, but it seems to actively disorganize activity within regions. In this case, this was an fMRI study, I believe, and this was looking at different networks within the brain. Networks, for instance, connecting the back of the brain to the front of the brain, or the two sides of the brain. And they were looking at the degree to which the activity in those two networks oscillated together or were completely opposite of each other or random with regard to each other. And they found that the higher the dosage, or when you're taking psilocybin, that the networks that were more distant from each other became more random with relevance to one another. So it's almost as if the communication between regions was really getting disrupted and randomized. All right. Now, the other thing that separates, we were talking about maybe the commonality between ketamine and something like LSD might be that it's cutting off communication between different distant parts of the brain. That might be something common. But another difference between those two hallucinogens is how it causes the brain to oscillate. So what these colorful maps here indicate are regions of the brain that are either turned off in purple and blue or turned on in orange after ingesting, in this case, psilocybin or ketamine down here. And here the different frequencies at which these regions of the brain are oscillating are indicated. Here we have beta band oscillations, 13 to 30 hertz, gamma oscillations, 30 to 50, high gamma. So these are really high frequency oscillations. And this is a result of psilocybin, or something very similar to LSD. Here you have ketamine also activating these beta band oscillations to a very high degree all over the brain, but doing the exact opposite with regard to these high frequency oscillations. So ketamine, even though you're also getting hallucinations, the brain is in a completely different state globally when it comes to these coordinated activities of many neurons at once, that you're getting these high frequency oscillations. And that's nice because it actually correlates with research that people in my lab are doing, including Matt over there, who's sitting at one of the tables. And he was doing, actually he was, I think he had to go, okay. So, and students in my lab doing research on the effects of ketamine and its potential involvement in reducing the motor pathologies associated with Parkinson's disease. We're trying to understand how ketamine can actually cause or alters brain circuitry. And so what we've done is we're doing this with rats, and we're trying to see in the efforts to find better treatments for Parkinson's disease whether or not it also induces these oscillations in animals. And we find the effect is really striking. And so in humans, they had to stop analyzing because their system wasn't capable of going above essentially these high frequencies of 100 times a second, 100 hertz. But we found in the rat that you can actually see oscillations induced by ketamine, and presumably the rat was kind of tripping out because he's kind of running in circles for a while, and so he's probably having some sort of experience there. But you see these really high intensity oscillations at around 150 hertz, 150 times a second, and this is time is here as on the x-axis. This is in minutes, and so you give an injection at the red line, and you can just see how the brain, in this case, the brain it's called the ventral striatum. It's an area involved in addiction and reward. How that area of the brain really starts to oscillate, to vibrate electrically at these super, super high frequencies. And it can last for hours. And what we think is that in treating things like Parkinson's disease or depression, what those oscillations might be doing is triggering drastic changes in brain networks that helps reset the system effectively to maybe to stop this aberrant learning that might affect or cause these tremors to develop in Parkinson's disease, or perhaps short circuit that recursive thinking that might contribute to depression and things like that. And so we're at least hypothesizing that's the case. We're dealing with rats right now, so it's hard for us to make big inferences from that. But we're definitely seeing a very similar response in rats, except at even higher frequencies than they do in humans with ketamine. All right, but what's interesting, and I think goes back to this whole isolationist hypothesis, is that if you go and look more closely at different regions of the brain and their connections to each other during these oscillations, that you can see that certain oscillations, those high frequency oscillations, this just indicates how strong two brain regions interact with each other at a high frequency, the blue line. And each vertical line indicates a ketamine injection. And you see that at these high frequencies, these regions of the brain interact really strongly after each injection at high frequencies. But at lower frequencies, these regions of the brain become almost completely decoupled from one another. And so it does complicate the story a little more, but we're thinking that ketamine might actually cause increased connectivity between regions at really high oscillatory frequencies. That means the neurons are communicating at a really high rate, maybe in a very disorganized way. But the low frequency communication might be turned off or shut down as a function of ketamine. So there's this anti-relationship essentially between the high frequencies induced by ketamine and then these low frequencies that are induced by ketamine. All right. All right. So to summarize, to put it all together, LSD and ketamine produce really powerful hallucinations. But how does that happen? These are very, very different chemicals, very different drugs, and they activate very different systems. But some commonality between these two regions, for example, is turning off a region like the thalamus, these hubs that connect many different regions of the brain together. And again, the brain, once brain regions are left to their own devices, they make stuff up. And so that may be one reason why disconnecting different brain regions from each other results in hallucinations. But you can't argue that these different drugs have very different subjective effects, at least on the self-reports that you get from people. And so that may be due to the very, very different patterns of electrical activity induced by those two different drugs, because they are actually activating different regions of the brains, and they're activating them at different frequencies. And so that might contribute to the different subjective effects of these two different drugs on the brain. All right, so that's the review of the brain and oscillations and the neural basis of hallucinations. So I'll be here for quite a while. So if anybody has any questions, the microphone will start being passed around. Thank you very much.